Lecture for Chapter 5, Microbial Metabolism, Part 1. Metabolism can be defined as the sum of all biochemical reactions within a living organism. And all of these reactions will involve organic molecules, otherwise known as the macromolecules. So before I start talking about metabolism itself, I want to give you a review on organic molecules. First of all, organic molecules are based on hydrocarbon skeletons. And hydrocarbon skeletons means that they are composed of hydrogen and carbon. And so all the different types of organic molecules have this basic skeleton structure. And they can vary in length. They can have single or double bonds. They can be branched or unbranched, and they can also be arranged in rings. So with these hydrocarbon skeletons, you get a lot of variation in the structure of the organic molecules. In addition to the hydrocarbon skeletons, these molecules also have various functional groups. And these functional groups are groupings of atoms that are not just carbon and hydrogen. So there are hydroxyl groups, there are carbonyl groups, amino groups, carboxyl groups, for an example of a few. And these different types of functional groups have different types of characteristics which give the different organic molecules their different functions. Because we are talking about molecules and reactions, I want to go over the two types of chemical bonds that are most commonly associated with living things. The first are covalent bonds. And covalent bonds, co stands for together, like cooperate or coordinate, and valent refers to valence electrons. So hopefully in chemistry you learned about atomic structure. All atoms have protons and neutrons in the nucleus, and they have electrons orbiting around the nuclei. So those electrons that are in the outer shell, those are called the valence electrons. And for a covalent bond to form, that means that two atoms will be sharing valence electrons. So the two atoms will share those valence electrons, so that pair of electrons will be orbiting both nuclei. So this would be one covalent bond. And here's another pair of valence electrons being shared between these two nuclei, and those valence electrons will orbit the two nuclei. And you should know that covalent bonds are very strong, very difficult to break, and they are depicted by straight lines. So in a molecule of water, you have two covalent bonds, one between one hydrogen and the oxygen, the other between the other hydrogen and the oxygen. The other type of bond are hydrogen bonds, and hydrogen bonds are also very important to living things. And hydrogen bonds are not a typical bond like a covalent bond, but it is an attraction between a partial negative charge and a partial positive charge. And it's always depicted by these dotted lines. And hydrogen bonds, by their characteristics, they're very easily formed and broken, very easily formed and broken. So individual hydrogen bonds are not very strong. They're very weak, easily broken and formed again. But when you get a lot of hydrogen bonds together, you get a very strong force. The four types of organic molecules that make up all living things are the carbohydrates, the lipids, the proteins, and the nucleic acids. And sometimes these organic molecules are referred to as macromolecules because in the molecular world, they're very, very large molecules. And for the most part, most of these organic molecules, most of these macromolecules are composed of similar subunits that are called monomers. And those monomers are linked together by covalent bonds, sharing of those valence electrons, to form large molecules called polymers. So most of these organic molecules have that basic construction. They're linking together similar subunits called monomers to form the large polymer, the large macromolecules. The first type of macromolecule or organic molecule that I want to review are the carbohydrates. And the name carbohydrate tells you the two types of atoms that are found mainly in the carbohydrates, and that's carbon and hydrogen. So those are the two main atoms that you find making up carbohydrates. And carbohydrates, their main purpose is to provide energy for the cell, readily usable energy. And some carbohydrates are also involved in structure. The basic monomer that makes up the carbohydrates are the monosaccharides. So monosaccharides are also referred to as the simple sugars. And this is an example of the structure of glucose. And glucose is also known as blood sugar. Any word that ends in ose is usually some sort of sugar. So fructose, lactose, all of those are sugars. You can take these simple monosaccharide structures, link them together with covalent bonds, and that forms the polysaccharides. Polysaccharides, many sugars linked together. And some examples of these are starch, glycogen, and cellulose. So again, carbohydrates are made mainly of carbon and hydrogen with some oxygen thrown in, and their main purpose is energy, especially those simple sugars like glucose, and you can link simple sugars together to get polysaccharides like starch, glycogen, and cellulose. Next are the lipids, and the main characteristic of lipids is that they tend to be hydrophobic. Hydrophobic, they do not dissolve in water. You can also refer to them as nonpolar. Also, again, they do not form hydrogen bonds with water, so they don't dissolve in water. And in terms of function, lipids are often used for long-term energy storage in a cell. And they also have some other functions too. Often the most common lipid that is described is a triglyceride. Triglyceride is composed of one glycerol, which is a 3-carbon alcohol, which is then bound to 3 fatty acids. So this is a fatty acid here. And fatty acids can either be saturated or unsaturated. A saturated fatty acid has only single bonds between all the carbons in the fatty acid. And an unsaturated fatty acid will have at least one double bond between the carbons, and that often creates a kink because a double bond is shorter and more rigid than a single bond. And so you can have, this is a mono unsaturated, you can also have poly unsaturated fatty acids, and a poly, meaning many, would have more than one double bond. Another example of a lipid is a phospholipid. So you should know that a phospholipid is the type of lipid that makes up the cell membranes. So some lipids are involved in structure. And the phospholipid has a phosphate group and it has 2 fatty acids. So these are the 2 fatty acids. So it has the basic structure of a triglyceride, except it only has 2 fatty acids and the other group is a phosphate group. And this creates the hydrophilic head, which interacts with water, and then the 2 fatty acids are hydrophobic tails, which do not interact with water. Then the last example are steroids. So some steroids are lipids, and one example is cholesterol. Cholesterol is a nonpolar lipid, so it does not dissolve well in water. And some other examples of steroids that can be derived from cholesterol are progesterone, aldosterone, testosterone, cortisol, and estradiol. Proteins are the next group of organic molecules, and proteins are really important because they have many different types of functions. And this diagram shows you the different functions that they can have. First is structure, so they are important for the structure of individual cells, like the fibers in the cytoskeleton, and they're also important for structure in terms of muscle tissue in multicellular organisms. And again, with the muscle tissue, also movement. Also, proteins are incredibly important for cells because they serve as enzymes. They catalyze different biochemical reactions. And in multicellular organisms, they serve as hormones, like some of the lipids. So they are molecules of information. They can communicate between different cells in a multicellular organism. Antibodies are also composed of proteins, and at the end of the semester, I will be going over antibodies. Also, some important functions, especially in your body, is fluid balance, acid-base balance, membrane transport for your cells, and also transport of different molecules in your blood. So proteins have a wide variety of functions, which makes them a little bit different than the carbohydrates and the lipids. Protein structure is a little bit more complicated compared to the other organic molecules. And this is the reason why we get so many more different types of functions for proteins compared to the other macromolecules. The basic subunit for protein structure are amino acids. So the amino acids are the monomers that are linked together to form proteins. And every amino acid has a central carbon, which is covalently bonded to a hydrogen. Then another covalent bond is to an amino group. Another one is to a carboxyl group. And the last covalent bond is to a side group. And this is like a functional group. And in living things, there are 20 different potential side groups. So in living things, there are 20 different types of amino acids that can be linked together. And each amino acid has its own unique functional group, so it has its own unique characteristics and function. When talking about protein structure, the three-dimensional structure is really important to its function. And protein structure has 3 levels of physical structure. The first level is called the primary structure, and the primary structure is just the order of nucleotides. So the order of nucleotides and how they are covalently bonded together. Secondary structure has to do with how this initial chain, polypeptide, starts to fold on itself. And this happens when you have that polypeptide in water. You can get 2 types of folds, an alpha helix, which is kind of like a corkscrew shape, or a beta pleated sheet, which is sort of like when you fold paper with a fan fold. Both of these are held together by hydrogen bonds. So you can see these little hydrogen bonds, the dotted lines are holding the alpha helix together, and hydrogen bonds between the beta pleated sheet holding that together. So secondary structure in proteins is dependent on hydrogen bonding. The next level of structure is tertiary structure. So this would be like the 3-dimensional structure, 3D structure. And this is how that whole polypeptide structure forms a 3-dimensional structure. And again, that is also dependent on hydrogen bonding. And then the last level of structure is quaternary structure, and that's how this individual protein, this polypeptide, interacts with other proteins, other polypeptides, to form a functional protein. So that could be your enzyme, or that could be your hormone. The last type of organic molecule that you find in cells are nucleic acids, and nucleic acids are the molecule of information for the cell. And I went through nucleic acids in a lot more detail in the genetics lecture, but just as a quick review, the 2 major types of nucleic acids you find in cells are DNA and RNA. And some differences between the 2: DNA is a double-stranded helix, RNA is single-stranded. The type of sugar you find in the nucleotides in DNA is deoxyribose. In RNA, it is ribose. And then the nitrogenous bases that you find in the nucleotides are slightly different also. DNA has adenine, guanine, cytosine, and thymine. RNA has adenine, guanine, cytosine, and uracil in the place of thymine. And both of these molecules can perform complementary base pairing. In DNA, adenine pairs with thymine, guanine pairs with cytosine, and in RNA, adenine pairs with uracil, and guanine pairs with the cytosine. And of course, these are very involved in DNA replication and gene expression, especially in the process of transcription and translation. As I mentioned before, metabolism can be defined as the sum of all chemical reactions that occur within a living organism. And this could be the whole multicellular organism like a human, or it could be referring to an individual cell. But for any organism to be considered a vegetative, actively functioning organism, they have to be metabolizing. And this is an example of some of the biochemical reactions that are known that occur in an individual cell. And again, as I mentioned, all of these biochemical reactions involve macromolecules. And in this diagram, they are divided by color. So these blue colors, those are the pathways, the biochemical reactions that are involved in carbohydrate metabolism. These teal colors have to do with lipid metabolism. The orange ones relate to amino acids, which are the building blocks of the proteins. So some up there, some in the darker orange. And then the red are the nucleotide related metabolic pathways. So that has to do with the nucleic acids. And usually when they make these maps of the different types of biochemical reactions in a cell, sort of the central idea is the carbohydrate metabolism. That is the major pathway that generates energy. And often when you look at this, if you see a circular pathway, that is the Krebs cycle or the citric acid cycle, and that is led into by glycolysis. So this is just an example of some of the biochemical pathways that occur in an individual cell. And if you look at them, a lot of them are interrelated. So you can start with a lipid metabolism, and you can find a pathway which eventually leads you to produce nucleic acids. This figure shows you in a little bit more detail the metabolic processes that surround carbohydrate metabolism. So one of the most important purposes of carbohydrate metabolism is to produce ATP, which is the usable, readily usable energy for the cell. So carbohydrate metabolism starts here. So this starts with the carbohydrates like the starch, and eventually all these different carbohydrates can be broken down into glucose. And then glucose will go through glycolysis. Once it goes through glycolysis, forming pyruvate, then that can enter into the citric acid cycle and eventually go to the electron transport chain where ATP is produced. And ATP is such an important product. All living cells need a constant supply of ATP because that's the readily usable energy for the cell, that if carbohydrates are not available to the cell, they can break down other macromolecules to produce this ATP. So one example are the fats. The fats can enter the pathway this way, or the fatty acids can enter through a slightly different way. If fat is not available, then they can break down proteins. There are all sorts of areas here with the different amino acids, how those can start enter the pathway to be broken down. And even if there aren't any proteins available, some of the nucleotides, the purines and the pyrimidines, can also be broken down. Also with this is that these pathways are not just one way. If a cell needs to produce purines, it can actually convert some of the sugars into purines. And even you can follow along, if it needs nucleotides or purines, it can take amino acids and they can go through this pathway and eventually produce purines. So these pathways are very important for the cell to control what it needs to build up and what it needs to break down. In general, there are two types of biochemical reactions: anabolic reactions and catabolic reactions. And when thinking about those two different types of reactions, there are three aspects you need to keep in mind. The first thing you need to keep in mind is what is happening to the physical molecules. Are you building molecules up, making large molecules, or are you breaking them down to make smaller molecules? Second thing you have to keep in mind is water. How is water involved in the reactions? And the last thing to keep in mind is energy. Are you storing energy in these reactions, or are you releasing energy in these reactions? The first type of molecule to discuss are the anabolic reactions. And when I go through this, remember to pay attention to what's happening to the molecules, how water is involved, and what happens to the energy. This is a diagram of an anabolic reaction, and often anabolic reactions are referred to as dehydration synthesis reactions. In terms of the molecules, in an anabolic reaction, you are forming a larger molecule. So again, these organic molecules, most of them are made with linking together monomers together to form a polymer. And in this case, you're taking a molecule of 2 monomers and you are adding a 3rd monomer. So the result is going to be a polymer with those 3 monomers bound together. In terms of water, dehydration synthesis reactions result in a molecule of water being formed. And that is because in order to link these 2 monomers together, you have to break pre-existing covalent bonds, because that is going to free up electrons so that these 2 monomers can then form a new covalent bond together. So in order to link them together, you need to break 2 pre-existing covalent bonds. So one subunit is going to release a hydrogen, one is going to release a hydroxyl, and then those 2 will come together to form a molecule of water. So that's a dehydration synthesis reaction. In dehydration, you are removing water. The last thing to think about is energy, and in general, anabolic reactions are endergonic. Endergonic, and that means that you have a net input of energy. So this is going to take more energy to link these monomers together than is going to be released. So this is going to store energy. You're going to have a net input of energy into this type of reaction. The second type of reaction are catabolic reactions. And this is an example of a catabolic reaction. And catabolic reactions are exactly the opposite of anabolic reactions. And catabolic reactions are often referred to as hydrolytic reactions. So in terms of the molecules, what is happening in a catabolic reaction? You are taking a polymer and breaking it down into smaller molecules. So in this case, you're breaking off that subunit. And in order to do this, you are going to lyse or split a molecule of water, and that's hydrolytic reaction. Lytic means to split. So what is basically happening is that in order to separate off the subunit, you will have to break that covalent bond. And once that covalent bond is broken, molecules don't like unpaired electrons, so you're going to have to make some covalent bond up in that place. So a molecule of water will be split, and one of the hydrogens will form the new covalent bond to one subunit, and the hydroxyl will form a covalent bond to the other subunit. And so this is a hydrolytic reaction. And catabolic reactions in general tend to be exergonic. Exergonic means that there is a net release of energy. So in catabolic reactions, usually you get a release of energy. So you're going to release more energy than you put into the reaction. When you look at the metabolism of a single cell, all the biochemical reactions that are occurring in that cell can be grouped together as either catabolic reactions or anabolic reactions. So the catabolic reactions are basically the reactions where something is being broken down and energy is being released. And anabolic reactions are basically divided into where things are being built or created and energy is being stored. It is very important for living things to be able to control all the different biochemical reactions that are occurring in the cell. And to really understand what is needed to control reactions, we need to think about basic chemical reactions. So in a basic chemistry class, most of the time in lab, all you are doing is performing reactions. And whether this reaction is in a chemistry lab or in a living cell, all reactions involve breaking covalent bonds. So you have a molecule, you need to break some of the covalent bonds, So then those molecules are free to form new covalent bonds to form some sort of new molecule. So essentially, for a biochemical reaction, in order for it to occur, there needs to be a breaking of covalent bonds. One of the basic ways to facilitate a reaction is to have two molecules collide with each other. If they hit with enough force, it may be possible to break pre-existing covalent bonds and allow those two reactants to reassemble and to form new covalent bonds to form products. So this is just an example of a reaction. You have two molecules that are coming in with enough speed that they can collide, and that will produce enough energy. They'll hit with strong enough force that it will break bonds and then create new bonds between the different molecules, forming different products. But with this, the two molecules need to hit with sufficient force to break those covalent bonds, and also they have to hit with the correct orientation. If these two molecules do not collide at the right orientation, they will not produce the products. So this type of reaction depends a lot on speed and orientation. This is often what you have in inorganic situations. To increase the probability of a reaction occurring and reactants being converted into products, you need to increase the probability that the reactants are going to interact with enough energy, enough speed, and in the correct orientation to form the products. There are two basic ways this can be done. First, by increasing the concentration of the reactants. So if you increase the concentration of the reactants, then there is a higher probability that the reactants will interact in the correct orientation and with the correct energy to form the products. So if you have very few reactants, you have a low probability of them interacting with enough speed and in the correct orientation. But if you increase that concentration, you increase the likelihood of the reaction occurring. Another way you can increase the likelihood of the reaction is by increasing the speed of the molecules. And to do this, you increase the average speed of the molecules by increasing temperature. So in this case, you have the same concentration of molecules, but now as you increase speed, they have more energy and they interact with each other more often. So you are increasing the likelihood that those reactants will be converted into products. Living cells have thousands of biochemical reactions occurring every minute. And they have to be able to have precise control over every reaction so they can respond to the environment. They can respond to a lack of nutrients, overabundance of nutrients. They can replicate. They can form endospores. So they need precise control over every reaction that is occurring. So the different possibilities, methods of increasing the rate of reaction, like increasing the concentration or increasing the temperature, are not viable options for the cell. The cell needs a more reliable and faster method of controlling their reactions. Because cells need precise control over thousands of biochemical reactions every minute, they use biological catalysts to control their reactions. And a catalyst is something that makes the reaction much more likely to occur. And the biological catalysts that cells use are enzymes. So these are protein molecules that catalyze the different biochemical reactions in a cell. In order for an enzyme to catalyze a reaction, it's going to do two things. The first thing it's going to do is to decrease the activation energy that is required for the reaction. The second thing it does is it holds the molecules in the correct orientation to facilitate the formation of reactants. So in terms of the activation energy, this is a graph that shows or relates the activation energy and what happens with an enzyme. So it begins with the reactants. This is the reactant, a molecule that's composed of two parts, A and B, And during the course of this reaction, it's going to form the products, a subunit A and subunit B. So this reaction looks like it's a catalytic reaction where it's taking a larger molecule and breaking it into its two subunits. For any type of reaction, there's a certain amount of activation energy that is required to destabilize the bonds. So destabilize those covalent bonds, and in this case, the covalent bonds that are holding the reactants together. So what an enzyme will do is it's going to destabilize those bonds, so you need less energy to go into the reaction to cause them to break. So that was a catalytic reaction. If this is a dehydration synthesis reaction, there is still a need for covalent bonds to be broken. So these covalent bonds need to be broken so that this subunit can then be linked by forming a new covalent bond between that subunit also. And also what enzymes are going to do is they will hold the two subunits in the correct orientation to facilitate this linking. So again, enzymes are going to lower the activation energy required for a reaction to occur. And the way they do this is destabilizing the covalent bonds. And they also hold the molecules in the correct orientation to facilitate forming the new correct covalent bond. The structure of an enzyme is very important to its function. Every enzyme has two important components. The first part is called the apoenzyme, and the apoenzyme is the larger part of the enzyme, and it is composed of protein. So this is the protein component of the enzyme. Every apoenzyme needs to bind to a coenzyme or a cofactor. So every enzyme is going to need two parts, an apoenzyme and a coenzyme or a cofactor. And the difference between a coenzyme and a cofactor is that a coenzyme is organic. It's a carbon-containing molecule, whereas a cofactor is inorganic. So it could be an ion, it could be a metal, it's some other inorganic molecule. But in order to form the holoenzyme, the whole active enzyme, you need to have your apoenzyme bound to some sort of coenzyme or cofactor. And this enzyme, or this holoenzyme, is now active because now it has formed the appropriate active site. And the active site is where the substrate will fit perfectly. A lot of books say lock and key. I think of it as puzzle pieces, but you need the combination of this coenzyme cofactor plus the apoenzyme to create the appropriate active site. And if you notice, the apoenzyme by itself does not have the appropriate active site, and the coenzyme or cofactor does not have a complete active site. You need both of these to bind together to get that complete active site by which it will bind the substrate. It is very important to understand how enzymes catalyze reactions. This is a figure that goes over the basic steps of how an enzyme can facilitate converting a substrate into a product. So this is the holoenzyme, and every holoenzyme has an active site. And the active site has a particular shape that will fit only one type of substrate. So only this type of substrate will be able to be recognized by the enzyme, and the enzyme can only bind to that substrate. When the enzyme binds the substrate, it's going to form the enzyme substrate complex. And also during this time, you have something called induced fit that will occur. Induced fit. An induced fit is when you have the enzyme, so here is the enzyme binding the substrate perfectly in the active site. So this is the substrate that binds in the active site, and between the enzyme and substrate there will be hydrogen bonds forming. So remember, hydrogen bonds are denoted by dotted lines. So at all that interface between the enzyme and the substrate, there will be hydrogen bonds forming. And remember, hydrogen bonds individually are very weak, but when you get a lot of them together, you get a strong force. And so because so many hydrogen bonds are forming all over this interface, it becomes a very strong force that will cause the covalent bonds in the substrate to break and reform. So all of those hydrogen bonds all over here are creating such a strong force, it's causing the covalent bonds in the substrate to break and reform. And that is what is causing the reactant to be converted to the product. So here's a break in the product. And now, because by definition the products are different than the reactants or the substrate, this no longer fits perfectly into the active site, so they are released, and the enzyme is unaffected. So catalyzing a reaction is going to have no effect on the enzyme. Enzyme still retains its original shape, so it can start over. If there's another available substrate, it can go through the process again. So the enzyme is going to bind to the substrate that fits perfectly into the active site. Then once it does that, it's going to perform induced fit, where all of these hydrogen bonds form between the substrate and the enzyme. That's going to put stress on the covalent bonds in the substrate, which will result in the production of the products. Products will be released, enzyme is unaltered, enzyme can do it again. Induced fit is very important for the function of the enzyme. If the enzyme cannot perform induced fit, then it will be unable to catalyze the reaction. This is an example of a computer model of an actual enzyme. And enzymes are proteins, macromolecules, so they're much, much larger than substrates. And this is the active site, which will be able to bind to the substrate, and the substrate will fit perfectly into that active site. When the enzyme binds to the substrate, then those hydrogen bonds are going to form all over the interface of where the substrate and enzyme come in contact. And in fact, those hydrogen bonds are going to be so strong, it's going to cause the enzyme to sort of curl in around, kind of like grabbing hold of the substrate more. And so all of these hydrogen bonds are going to cause a really strong force, cause a lot of stress on the substrate, which will result in covalent bonds being broken and reformed in the substrate, converting the substrate into the product.